bring their band along.
Yeah.
They're supposed to have a really good band, Bethune-Cookman. Yeah, one of the best bands in the country.
Really?
Yeah, yeah, and last week, I don't know if you noticed, anybody go to the game last week? No. Minnesota brought their band, so they had two bands.
I guess our band is just very nice to like other bands and stuff.
I don't know.
They take the other band out and do stuff.
I don't know.
Yeah. Just
How
very
about our
cool.
volleyball team? Are we gonna win a match here anytime soon? Lost 3 in a row, right? 4 in a row, something like that. You all are too busy with school to do sports, right? I don't blame you.
We'll see if Scott Frost can keep.
What about the World Series? Is Boston gonna win?
Dodgers.
Dodgers? That's who I'm hoping for, but it doesn't look good right now.
I am hoping too.
My wife is a big Boston fan though. So, Nobody else has an opinion on World Series? No?
So it's Boston.
Yeah, it's Boston, Los Angeles, Dodgers. Okay. All right. No questions? Let's start. I'll just go through these, and as far as I get, as far as I get. Right? Going through and reviewing and emphasizing the points I think are important. So, a quick run through. Ready? Okay, relative water content. That's the relative to how much water there could be in a cell or a tissue or a plant. Relative to how much, the amount of water relative to how much there could be if that system were in equilibrium with the total water potential of 0. Kind of the maximum amount of water. Okay. Thermocouple. Okay, so the important instruments for measuring water potential, the two most commonly used are psychrometers and pressure chamber. And they both can measure total water potentials. Also, Shardakoff method, important, and pressure probe, yeah, kind of esoteric, something that people use. So, thermocouple psychrometer. First, what a thermocouple is, we don't have to go into all this. The important thing is the thermocouple measures temperature. It can be used to measure temperature. Or, putting it in a better way, a thermocouple has an output that's proportional to its temperature.
Would we need to know how the psychrometer works? Thermocouple?
In a general sense, yes. But the specifics, no. I mean, I'd like somebody to explain to me how a thermocouple works, because I don't know. All I know is it has an output that's proportional to its temperature. Why that happens is over my head. And you know, if my physics grade didn't decrease every semester that I took physics, maybe I would have stuck with it long enough to figure it out, but I never got that far.
I got a bachelor's degree in physics and I don't know.
Well, you're way more advanced than I am because I quit long before. I had a year of physics, college physics at Michigan State, but they were on a quarter system, so it was 3 courses. It was Newtonian mechanics was the first one, I guess. And what was the second one? I can't even remember.
Was that your graduate school or?
No, it was undergrad at Michigan State. I was a botany major at Michigan State, and since that was in a college of natural sciences, I was required to take a year of physics, a year of calculus, a year of inorganic chemistry, and a year of organic chemistry for a bachelor's degree in botany. So, I'm all for it.
My undergrad was pretty similar. Our plant biology and biotech is pretty similar to that here.
Here?
Yeah. It's
Good. As well it should be,
It's brutal.
if you ask me. I mean, Who asked me though? Nobody asked me. So, anyway, what was it? Newtonian mechanics. What comes next? Oh, electricity and magnetism. How can you forget that? Yeah, and then it was called waves, but it was kind of optics. And then the next one would have been statistical mechanics, which maybe would have started to get into that subject. I don't know. Like I said, every quarter my grade got worse and worse. So I cut my losses. But I still am really interested in physics. One of these days I'm going to work my way through those Feynman books. Anyway, after I retire, probably.
Whenever you have time.
Yeah, after I retire, that's what I say. So as far as how a psychrometer works, in a general sense, yes. And here's the general sense that I mean. How it works is we have a small volume, small chamber, and we put a sample in there. And then we let the sample equilibrate with the air in the chamber. We let the water in the sample equilibrate with the water in the air. You know, so that the water potential of the air is equal to the water potential of the sample. Then we need a device, some thing inside this small volume that can give us an indication of what the water potential of the air is. So the water potential of the air is given by this equation, right? R2 to the log of the relative humidity, or the vapor pressure divided by the saturation rate, the relative humidity. So this is the part that varies with this. So we need a thing in there that gives us an output that's proportional to the relative humidity in the chamber. And a thermocouple, if we have a thermocouple in there, well, the temperature is just the temperature, that's not a big deal. But if we have a thermocouple that we have a droplet of water on, that's evaporating, that evaporation rate is proportional to the relative humidity in the chamber. And what's more, that evaporation rate from the thermocouple determines the temperature of the thermocouple. It's like the wet bulb and dry bulb thing in a psychrometer. That's why this is called a psychrometer. So that evaporation rate is proportional to the relative humidity, and that evaporation rate determines the temperature, which is the output of our device. And so that's why a thermocouple can be used. We don't have to use a thermocouple in this device. And in fact, most psychrometers, or most instruments for measuring water potential in such a manner these days typically don't use a thermocouple. They use a chilled mirror. So that's the thing, you put a mirror in there and shine a light on it and look at the reflection from the mirror, and then you cool the mirror off. And when the mirror gets to the dew point, condensation on it interrupts that reflection, and you can get a signal from your device of what the dew point is. And the dew point is also proportional to the relative humidity inside the chamber. So the thermocouple is just a way to do it. But the overall idea of the psychrometer is that you let a sample equilibrate in a space, and then you have some indication of what the relative humidity is in that space. And that relative humidity varies as the water potential varies. So to use such a device, it's especially for a thermocouple psychrometer, we need to calibrate it against solutions that we know the water potential of. And a solution, a piece of filter paper dipped in a solution, the water potential of that water on the paper is its osmotic potential. There's no pressure component involved. And so we can use Van 't Hoff equation to make a series of solutions that we know the water potential of, and use those to calibrate or to check our instrument. But okay, this is showing that it's a sensitive thing. Here's the idea of the dry bulb and the wet bulb, relative humidity. Okay. So this slide summarizes what I would hope that everyone would take home from this idea of thermocouple cyclone, how it works. This is the depth of how it works that I would expect somebody to remember. Okay, and this is just a calibration relation, blah, blah, blah. Okay, but there are different ways, you know, different types of cyclometers have different ways of getting this output. The most the one for purists is this isopiistic cyclometer, where you put a solution on a droplet of a solution on the thermocouple junction, and you look for that solution that gives you no output from your device. In fact, no water is evaporated because the solution has the same water potential as the air. So don't worry about the different types of cyclometers. That's just a complication that we don't have to worry about. Right? Okay, so yeah, that's cyclometers, right? Any questions about that stuff? Other questions? Okay, so okay, here's another good summary slide of how a thermocouple cyclometer works. Okay. Precautions, so the geometrical considerations, epidermal resistance, absorption of water, heat of respiration. The most important consideration for using a cyclometer isn't the accuracy of the machine. It's not some of these other rather esoteric considerations. The most important factor to consider is loss of water between excising the tissue and putting it into the cyclometer. Because if you lose water, the water potential changes. There's a one-to-one correspondence between water content and water potential. And any water loss is going to give you a different water potential than the tissue had before you excised it. So special precautions have to be taken to minimize water loss when making these kinds of measurements. Okay, so that's typically, or cyclometers can be used to measure total water potentials. And a lot of times for plant studies, those are leaf discs. Just a punch from a leaf or a couple of punches from a leaf, you put it in a sample chamber. But we can also use it to measure osmotic potentials. And the cyclometer is measuring a total potential, but to measure an osmotic potential, we take a leaf or whatever tissue we're interested in, freeze it, thaw it, and squeeze the juice out of it, right? And wet a piece of filter paper with that stuff and put that in the cyclometer. And that gives an osmotic potential, because when you freeze and thaw the tissue, you disrupt all the cell walls and membranes, and all the stuff inside the cells comes out, and you get access to that for your osmotic potential measurement. The thing you have to watch out for is when you do that, you're mixing the symplast and the apoplast. And remember, in the symplast, the osmotic potentials are quite negative. There's a lot of solutes dissolved in the stuff in the cytoplasm, or in the cytoplasm of the cell. There's a lot of solutes in there. So its osmotic potential is quite negative. The apoplastic water has an osmotic potential close to 0. There's not much stuff dissolved in there. And so when you mix those two, you change. If you're interested in the osmotic potential inside these living cells, have to correct for the amount of water that you're mixing in from the apoplast. Apoplastic correction, and it's a simple thing to do mathematically. Beta is the apoplastic fraction, and you take your measured value, 1 minus beta, divided by 1 minus beta to get a corrected value. This is assuming that the apoplast has an osmotic potential of 0, which is not a bad assumption. It's pretty close to 0. In fact, the error that arises from assuming the apoplastic fraction has an osmotic potential of 0 is much smaller than the errors inherent in estimating this apoplastic fraction itself. It'd be nice if we could ignore this, but you can see from these numbers that the apoplastic fraction can be a significant amount of the total water in a leaf, let's say. 40% eucalyptus, 30% wheat. You see some of these other. So it's not, although I hesitate to give a number. You know, by far, the vast majority of people that measure osmotic potentials with a psychrometer ignore this. But I'm just saying, it's not so easy. I mean, you know, you shouldn't ignore it. You shouldn't ignore it. The problem is, you know, how can you estimate that apoplastic fraction? How do you know what that is? Well, now you know that from a pressure volume curve, you can get an estimate of that. But that's for a leaf. And if you're using a root, good luck. Or who wants to do a pressure volume curve? That's a time sink. It's a tedious measurement to make. And so usually people ignore this idea. But you're all smarter than that, huh? Okay. We can have an in situ psychrometer, measures total water potential of an intact leaf. We can have a psychrometer that's made for burying in the soil and to get a soil water potential, and so forth. Okay, that's psychrometers in a nutshell. That's what I think is important to remember. All right, so Shardakoff method. This is a nice technique that hardly anybody uses, but I like it because it illustrates well, I like it because if you understand how this works, then I think you're getting a good understanding of plant-water relations, you know, as a whole. So if you can explain to somebody how to do this and why it works, I think you are the expert in plant-water relations. Remember how it works. We make a series of solutions of known osmotic potential, known total water potential, and use something like sucrose as a solute so that it won't enter the cells. Reflection coefficient near 1. We take our sample, cut it up into little bits, and have 5 groups of those samples. Put it in a small container and introduce one of the solutions into each of those containers. So what happens, and then let things come into equilibrium. Let the water potential, the total water potential in the cells in the sample come into equilibrium with the total water potential of the solution. There's a membrane around these cells, and so it's the total potentials that are the driving force for moving the water. But that's what we want. If the sample has a total water potential more negative than the solution, water will go from the solution into the sample. What that does is it concentrates the solution that's left behind, because we've used sucrose that can't readily enter those cells. So it concentrates the solution. If the solution is more concentrated, it's more dense. So what we're going to do is reintroduce a droplet of this solution into the original test tube of our standard and see whether it floats or sinks. Before we can do that, we have to use some dye so we can see what's happening. We introduce it. If it sinks, okay, so in this example, our leaf had a total water potential of -1 megapascal. So when we put it in the 0.9, was it? Yeah. Yeah. If we put it in 0.9, it sinks. If we put it in the 1.1, it rises. If we put it in the 1, it stays there, right there. And so we know our sample had a total water potential of -1. Right? That's Shardakoff. As I say, people don't use it very much. The biggest precaution is, again, water loss from the tissue after you cut it from the plant and before you get to your final result. And any water loss changes its water potential, so you have to minimize that. So, not many people use it, probably because not many people are familiar with it. Doesn't take expensive equipment, doesn't take a lot of careful, well, it doesn't take a lot of, you know, it's not too, you don't have to spend months developing your technique before you can use it, that's what I'm trying to say. It's kind of a simple procedure.
So is there a reason why people don't use it?
Yeah, because I think because they don't know about it. Did you ever hear of it before? No. Before I taught this class, I never heard of it before. And the only place I've seen it is in this book chapter of Ted Chow in the Irrigation of Ag Crops book. He gives it a couple of pages in this review chapter of measurements. And he uses it, in fact, in his research, but that's the only place I know of it. And the reference that he cites for the technique is in Russian. Some journal in Russia, you know, published in Russian, so. Anyway. Maybe in Russia it's used a lot, I don't know. Could be. I don't know. Okay, I mean, first of all, not many people are interested in water potentials anyway. You know, the whole universe of people. Who's interested in water potentials? You know, so you quickly get to the small. Okay, but that's Shardakoff method. And then pressure probe is the way to measure pressure potentials directly by sticking a hollow needle right into a cell. And that's shown here. And what happens is you poke this capillary into the cell, and because the pressure potential in the cell is higher than the atmospheric pressure in a living cell, typically it's either at atmospheric pressure if it's at incipient plasmolysis, or if it's not, then it's higher than atmospheric pressure. So when you poke it, the stuff inside is going to squirt out. And what it does is it squirts out into this capillary, which is filled with oil, and moves this oil-water interface back into the capillary somewhere. So what the device does, you increase the pressure on this oil and force this back to where it was. And then however much pressure it took to do that, you know, is the pressure inside that cell. So it's a good technique, but you know, it's one cell at a time. And so we're doing an experiment. You have to have clearly defined objectives if you're going to go into the trouble of using a device like this. You also have to build it yourself, or you can't buy it. It's used quite often, I mean, as far as these things go. And, okay, so instead of using a pressure probe, which this fellow Shackle did, and compared different techniques. And remember, the results he got showed that when the leaf was transpiring, when it was not transpiring, 0 transpiration rate, the errors amongst the in situ cycrometer, the pressure probe, and the nanoliter osmometer, measure osmotic potentials, were about 0.05 megapascal. So, you know, they kind of agree pretty well with each other. But most people won't go to the trouble to use a pressure probe. And so what most people do is measure the total potential with a psychrometer, let's say, or a pressure chamber. Measure the osmotic potential with a psychrometer on frozen and thawed tissue, and calculate the pressure potential from the difference of those two. Not entirely satisfying, you know, because any error in this or this will be implicit in this, and you just don't know them. But as Shackle showed, you know, if you're careful, it's not too bad. Okay?
Just one question, for the Shardakoff
Yes.
technique,
Yes.
you can use any part of the plant or is it specific to leaves?
No, any part of the plant. Yeah, any part. Just have to let it equilibrate, you know, and for a chunk of tissue, like a
Root.
root, well, it may take longer to equilibrate than a thin tissue like a leaf. But, no, in theory, it will work with any. Has to be made up of cells, though. You can't do it for soil. You can't use it to get soil water potentials because there's no cells. No membrane to exclude the solutes from the equilibration process. Okay, what's next? Take a break. Pressure chamber. Yeah, there we go. So pressure chamber, very simple device. You know, this guy Dixon back in the early 1900s, late 1800s, used it to extract stuff from plants that you can analyze. Now it's made out of steel, and it's commercially available. Here's an example. And the way it works is you take your sample, you put, let's say, a leaf. Most commonly, these are used for leaves or small shoots. So you put your sample inside the pressure chamber with the end that you cut off sticking out. You seal everything up, and you increase the pressure inside the chamber until you just see the stuff in the xylem come up to that cut end. That's that end point. That's typically the end point. And at that point, the total water potential in the mesophyll cells in this leaf is made up of its osmotic potential, its pressure potential, defined in the usual way relative to atmospheric pressure, and then this extra pressure that we're adding from the pressure chamber, this P. So at the end point, when we hold the pressure constant, the water in the cells in this leaf is in equilibrium with the water in the xylem, and you're looking at the end of that water right here at the cut end. So that water in the xylem out here has a total water potential of the osmotic component plus the pressure component. The pressure potential is 0 because this water is sitting at atmospheric pressure, it's not inside the chamber. And the osmotic potential is close to 0. In fact, we can assume that it's 0. And so the negative of the pressure in the chamber is equal to the total water potential of the volume average total water potential of those cells in the chamber. It's a clever idea, and it can be used for measuring total water potential. But if we make a pressure volume curve, we can get a lot of additional information. Precautions, blah, 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 all these things. The most important thing, again, is water loss from the tissue after you excise it from your plant and before you reach the end point. Before you reach that pressure where the water's just up to the cut end. Any water loss will change the water potential of the sample that you measure. Okay, leaks, water loss. Here's the pressure volume curve. Here's an example of water loss. Okay, as quickly as they could measure it in this experiment, remember 30 seconds, they already lost 0.6 of megapascal. Here's another example of water loss affecting values. Water loss inside the chamber, because as you increase the pressure in the chamber, the leaf heats up, the air heats up, and the difference between the vapor pressure and the airspace inside the leaf and the vapor pressure of the air in the chamber gets bigger and bigger and bigger, and that increases any evaporation that you have from it. So that's a no-no. Exclusion errors and anatomical considerations. The biggest consideration is water loss. Moving right along. Pressure volume curves. Well, you all did this just recently, so we don't have to spend a lot of time on it, maybe. All right, but the pressure chamber measures total water potential. And typically, or a lot of times we're interested in I have my experiment and I have a treatment and I want to know what's the water potential of that leaf on that plant, you know, given what I did to it, right? And so you cut the leaf off the plant, put it in the chamber, find out what the water potential is. But we don't have to just do that. We can let that leaf from our plant come into equilibrium with the pressure in the chamber at any pressure. Especially, well, to get the water potential as close to zero, we have to hydrate the leaf. You know, have to let it take up water until it's really almost fully hydrated to get those values here. But we can do it, and we can stop at any point, at any pressure, and let things equilibrate. And then, again, the total water potential of the cells in the leaf is given by the negative of the gauge pressure in our chamber. And so we can obtain a data set at a range of pressures and relative water contents. And you just went through the exercise of figuring out the relative water contents because it's hard, it's impossible to measure quickly enough to get the fully hydrated mass. So we had to calculate that. It's not on this one. Had to calculate that by extrapolating the first few pressure points. That allows us to calculate the relative water content. At high pressures, so if we plot 1 over P, the pressure, relative water content, in this case, high pressures are down here, the lower end of the 1 over P. And the way it's plotted here, the relative water content is high here and low over here. But at high enough pressures, the total water potential in the leaf is equal to the osmotic potential because the pressure potential has gone to zero. It's past the point of incipient plasmolysis. The pressure potential in those living cells is zero, and their total potential equals their osmotic potential. At that point, this relationship is a straight line. This is the total potential of those cells in the leaf, but it's just their osmotic potential because the pressure component is zero. That's this area. At pressures smaller than that, at relative water contents higher than that, there's a pressure component, and so we deviate from this straight line. You can convince yourself that should be a straight line by going through this algebra here that we went through before. But at high enough pressures, we'll deviate from the straight, or at high enough relative water contents, we deviate from the straight line because the total potential, which this curve represents, is made up of a pressure and an osmotic component, not just the osmotic. We can extrapolate this osmotic potential line to estimate the osmotic potential at full turgor, the apoplastic fraction. We can read off the osmotic potential at incipient plasmolysis, the relative water content at incipient plasmolysis. We can analyze these points and get an estimate of the pressure potential at each of these points by, we know the total potential, that's why these dots are here, and we can calculate the osmotic potential from this relationship. We can plot the pressure potential then against relative water content, and the slope of that relationship is an estimate of the volumetric modulus of elasticity of all the cell walls in that tissue. Okay. I think that's all there is, right? Okay. This one won't be on that exam. But this one may, this is review, right? This is photosynthesis. Some cartoon of photosynthesis. That's okay. Questions? We just did this, so it should be fresh in your memory. Roots. Okay. Interesting thing about the anatomy of the root tip, remember this is the most distal, less than 1 millimeter of the growing root. And the xylem becomes functional at the same place that the endodermis develops. So that's an interesting correlation. It's also the same place where the root hairs start to form. Interesting correlation, and the reason it's important is because the endodermis has the Casparian strips, and that is the endodermal cells are surrounded by this substance they call suberin, I think, and it's hydrophobic. Water can't go through it. And so that blocks apoplastic movement of water. So all of the water that's being taken up by the root is forced to go through a living cell. That means it has to cross the membrane into the cell, has to cross the membrane back out of the cell to get into the xylem. Membranes are, if you compare the hydraulic resistance of a membrane with the hydraulic resistance of a tube, like the xylem or the phloem, or the hydraulic resistance of a cell wall, you can convince yourself, and if not, then I can show you Park Nobel's book where he goes through the exercise. Convince yourself that the hydraulic resistance of an open tube is the smallest of those three features. The tube has the smallest resistance. The cell wall has the next smallest resistance. And by far, the highest resistance to water movement is crossing a membrane. So if you think of the soil, plant, atmosphere continuum, and let's avoid very, very dry soils, moderately moist soil, and a plant that has its stomata open and is transpiring. You think of the water movement from the soil into the root, up the xylem, into the leaf, out the leaf, through the open stomata to the atmosphere. The highest resistance in that pathway is
The membrane. The membrane.
the membrane. And where does water have to cross the membrane? Here in the root, at the endodermis. That represents the highest resistance in that whole pathway. As long as the stomata are open, that's the highest resistance in that whole pathway of water movement. If the stomata are closed, they're very efficient at stopping water movement. So that is probably the highest resistance. But when the stomata are open, the highest resistance in the whole system, soil, plant, atmosphere continuum, occurs here at the root endodermis, where water is forced across a membrane to get into the cell and again to get back out of the cell. Okay, here's apoplastics and plastic. Okay, other important things. The rate of water uptake is highest for the younger portions of the root system. And by younger, I don't mean that most distal millimeter, I mean the end few centimeters, 10 centimeters of the root. Here's another experiment showing the same thing. Although the rates of water uptake are highest at the end of the root, the older parts of the root still absorb water. And in fact, in this experiment, when the atmospheric demand was changed by changing the relative humidity so that the transpiration rate changed, that difference in transpiration was caused by higher uptake in the older parts of the root system, not in these young parts of the tip. Interesting feature. Yeah, okay, so drying soils. So water stress. So plants coping with drying soils. The problem with a drying soil is twofold. Number one, the soil water potential, and this is more and more negative as we go up here. The soil water potential depends on its water content. So as the soil dries out, the soil water potential gets more and more and more and more negative. So in order to take up water from that very negative value, the cells in the root have to have even lower water potential. Otherwise, the water is going to move out from the plant into the soil. And to do that, what choice do they have but to concentrate solutes in those cells in the root? That's the only thing the plant can control is the solute concentration. So to deal with that, they have to accumulate more and more solutes in those root cells. The other aspect of existing in a drying soil is as the soil dries out, the hydraulic conductivity of the soil also changes and gets smaller and smaller and smaller and smaller. So that to take up the same amount of water in a dry soil versus a moist soil, because the conductivity is so different, the difference in water potentials has to be even greater in the dry soil than in the moist soil to move an equivalent amount of water. So drying soils represent a very stressful situation with plants because their water potential decreases and the hydraulic conductivity of the soil decreases, and so the water movement is affected by the dry conditions. the other aspect of water stress in plants, besides what's going on in the soil, is what's going on in the atmosphere. And we have this equation that you remember from the psychrometer, and the water potential of the air depends on the relative humidity. And relative humidities vary depending on where you are. And they can represent very, very dry conditions. So temperature of, a moderate temperature, let's say, of 25 degrees C with a relative humidity of 60%, not unusual. Water potential of the air is minus 70 megapascals. Very dry compared to inside a living cell, we may be at 0, minus a half, minus 1 megapascal, maybe minus 2. Minus 3 is getting really dry for a cell. And so the air sitting at minus 70 is super dry, and it sucks out any water that it can quickly because that difference in water potentials is so big. For more extreme conditions, air temperature 35 degrees C, not unusual. RH of 15, the water potential minus 270 megapascals. So these plants have a really tough time. Not only is the soil dry, but the air is super dry. And water stress can be incurred by either one of those conditions, dry soil or dry atmosphere. We made measurements, I'll show you a graph in a week or two, of plants growing in a fen in central Saskatchewan, Canada. And they're growing in standing water. It's a fen, there's water all over the place. These plants grow in standing water. And when the vapor pressure deficit of the air increased, okay, in the morning they would start with their stomata wide open. But by 10 o'clock in the morning, the stomata would close. And it was because the water potential of the air, the vapor pressure of the air decreased enough that the water potential, the plant was losing too much water and it couldn't supply water fast enough to replace that transpirational loss, and so it closes stomata. These are plants growing in water, and they can't move water fast enough to keep up with that dry atmospheric demand. Okay. That's it. Okay. Sometimes I wonder if I should do this like speed skating thing at the beginning of it. You know, like next Tuesday's our exam, right? So next Thursday, maybe I take 8 of these PowerPoints and go through them really quickly. Think that's better or worse? I mean, I would go slower again, but I don't know. Maybe, you know, repetition is one of these teaching techniques you're supposed to use. Repetition. I don't know. Maybe we can try it. What do you think? You don't care?
It would give us like a full picture of the spectrum.
You know, it might work for a topic like stomata. There's a lot of stuff to think about and remember, and I'll consider it. I just thought of that now. Maybe we're moving really quickly through this material. It's kind of fun. We could cover a lot more ground if we wanted at this speed through the whole semester, right?
No, that's not a good idea.
Okay, xylem and phloem. Okay, they both move water, right? Or move fluids, let's say. Xylem, so the big thing about this is the cells that conduct the water are dead when they're functional. They have no membranes, no plastids, nothing. The components of the xylem tissue: tracheids, vessel members, fibers, parenchyma, tracheids and vessel members are responsible for moving water. And tracheids are found in gymnosperms and the so-called lower vascular plants. Here's some examples. They're connected. Each of these cells is a tracheid. And they have connections between one tracheid and the next. So here's one, here's another one. And these connections are called pits. They're open holes in the cell wall. I mean, the cell wall is kind of a mesh anyway. It's got holes all over it. But the pits are even bigger than those normal spaces between the cellulose microfibrils. They're a little bit bigger than that. Not a little, they're a lot bigger than that. And one tracheid is connected to the other through these pits. And what you wind up with then is a chain of cells that are connected that water can move through. The vessel members and angiosperms, a few gymnosperms like a are just elaborations of this idea. They are even, the connections are even bigger, right? So these are some vessel members from different species of mangroves. You know, some of them, they have different shapes, but some of them actually look like, you know, a piece of a pipe with a big hole in the end. And these things then are arranged end to end. These are called vessel members, and then the resulting chain of cells connected like this is a vessel. The vessels are connected to each other through pits, just like the tracheids. And what's more, some plants have tracheids and vessels, and they can be connected by pits. So it's a redundant system. And the important thing to remember is once water is in this vessel or string of tracheids, the driving force for the movement of that fluid is a difference in pressure potentials. There are no membranes involved once you're inside the xylem. Just like inside the conducting cells of the phloem, as we'll see. And so the importance of the governing equation, the first approximation for the movement of that water, is this Hagen-Queysui equation, where the driving force is this gradient in pressure potentials. Because my potential has nothing to do with it. Okay, so here's these things, speed of ascent. Here's our wood. Some trees have annual growth with vessels of the same size throughout. Some trees make large vessels early in the season, smaller vessels late in the season. The exact reason for that is an area of considerable research right now. And if you're interested in this, I can point you in some directions of, there's a lot that's known about this kind of stuff. But for our purposes here, let's just remember that the driving force for movement in the xylem is this gradient in pressure potential. And the amount that you can move for a given gradient is proportional to the square of the radius. So these big vessels here can move a lot more water than these little vessels here for a given potential difference. Okay, here's an even bigger, even more dramatic example. Okay, fibers are structural support, parenchyma, cells associated. All right, quote of the day, this was a couple of weeks ago. Negative pressures can only survive in the xylem of plants for long periods of time because water is confined to small compartments, tracheas and vessels. Because oftentimes that column of water is interrupted by a bubble of air. This happens in a couple of ways. When the water potential of the xylem becomes very, very negative, air can actually move in through a discontinuity in that column somewhere, in the cell wall or in the pit, through one of the pits or something, and make a bubble of air inside the vessel or inside the trachea. And that blocks off the water movement and increases the hydraulic resistance of the xylem. The other way that it can happen is in temperate climates, as we transition from autumn to winter, and the fluid inside the xylem freezes. Air has much lower solubility in ice than in water. And so as the water freezes, bubbles of air form. And those bubbles can block off these tracheas. Block off the vessel. So this formation of an air bubble is called cavitation. The formation, the bubble is sometimes called embolism. And these cavitation events are very common. Here we see in a maize plant, cumulative cavitations per day, 5,000, 10,000, 15,000 per day in a single plant. And you can see that in a droughted plant, you get a lot more of these than in a well-watered. So they're associated with low water potentials. In fact, here in woody plants shows different species, and as the water potential in the xylem gets more and more negative, these relative acoustic emissions increase. So these embolisms increase, and it affects the hydraulic conductivity or the hydraulic resistance of that system. So as the water potential gets lower, you get more and more of these cavitations. They block off more and more vessels, more and more tracheates, and the resistance increases. The loss of conductivity increases. This just shows 1, 2, 3, 4, 5, 6, 7 different species. Similar things are found all over the place. So this is a common occurrence in plants. You have to deal with it. Again, showing in a drying soil, as the water potential of the soil decreases, because the water content is decreasing, its potential becomes more negative. The water potentials in the plant also have to become more negative to keep taking up water. And as the water potential in the xylem becomes more and more negative, you risk blocking off the pathway with more and more of these air bubbles. So not only in a drying soil does the hydraulic conductivity of the soil decrease as the water content decreases, but the hydraulic conductivity of the xylem in the plant in equilibrium with that soil also decreases as the soil dries out. Okay, ordered pits. All right, and then this was just an interesting feature of xylem that even though the cells are dead, their conductivity can be affected by the composition of the fluid that's moving through. I'm gonna go esoteric. Okay. This one, for your own information, is isohydric and isohydric for your own information. But the bottom line, remember, was these aquaporins affect the hydraulic conductivity of the root system in these plants. So the expression of the genes that control the formation of aquaporins is a way that the plant has of altering the hydraulic conductivity of itself. And here it's the root system. Okay? Moving right along. What's that? Phloem. Okay, phloem, similar to xylem fibers, parenchymal cells, but the conducting cells are sieve cells or sieve 2 members, each one of which is associated with a companion cell that comes from the same mother cell. Mother cell divides, 1 cell, sieve cell, 1 cell, companion cell. unlike the xylem, the sieve cells and sieve tube members are alive when they're mature and functional. They have no nucleus, they have no organelles, and the reason they can stay alive is because of their associated companion cell right next to them. Keeping them going, let's say. Again, they're connected end to end by more or less elaborate holes in the end of the cell. Holes not covered with a membrane, totally open. So the membrane of this cell, the plasma membrane of this sieve element, would go along here. This is one of those pores in the plate at the end of the cell. The membrane would go along like this and be continuous over here into the next cell, but it does not block off this hole. So again, it's an open hole, and the driving force for water movement in the phloem is the gradient in pressure potential, not the osmotic potential, not the total potential. Pressure potential. Companion cells affiliated with the sieve elements, parenchymal cells. Unlike the xylem, phloem is a fairly concentrated solution. The most common solute in most plant species is sucrose. That sucrose is usually the product of recent photosynthesis. There's also a bunch of other things found in the phloem. But even though it's relative to the xylem, it's a concentrated solution, still the movement within the phloem, just like the xylem, is driven by differences in pressures. The osmotic component doesn't play a role. Even though in the phloem, typically, sucrose is moving from high concentration in the sources to low concentration in the sink, that concentrated sucrose doesn't play a role in the movement within the phloem. All right, aphids. Now, it's a little more subtle than that, and this was from last time. Here, let's go with this one. This one's a good one. Because although within the xylem, well, the fluid there is moving in response to pressure. So here's minus 0.5 megapascals, here's minus 0.7. Higher to lower potentials, the fluid moves this way. Within the phloem, pressure potential positive 0.6, positive 0.3, higher to lower pressure. Within the phloem, that's the way the water's moving, or the fluid is moving. Because the xylem and phloem are close to each other, water can move from one to the other, and that movement of water between the xylem and phloem involves crossing a membrane around these living sieve cells, sieve elements. And so now the movement between these two is driven by a difference in the total water potential. So in this example, we see total water potential minus 0.6 megapascals. Here, total water potential minus 0.4. Water is going to move from higher to lower potentials. On the other end of this system, total water potential minus 0.8. Here, total water potential minus 1.1 megapascals. Water moves from higher to lower water potentials. That sets up a circulation of water between the xylem and phloem, and that's Monk's pressure flow hypothesis, whatever he called it, for water movement in the phloem. By doing that, as a result of that, whatever is dissolved in the water moves the way the fluid is going. And so we get a high concentration of solutes in the source, low concentration in the sink, and that moves right along with the fluid. So it's an efficient way of transporting solutes from sources to sinks, as long as the plant can keep introducing solutes at this end and removing solutes at this end. And as long as that continues, then this circulation can continue and things can move around.
Is that why the water is so important for like reproductive stages of say like corn or something like that?
Yeah, well, that's part of it. The other thing with corn especially is the pollination, or the pollination of the silks from the tassel, right? The movement of that pollen, that's really sensitive to how dry the air is and stuff like that, and that can have a role to play in how many kernels how many kernels develop. The other thing I would say is because a lot of these solutes, or maybe almost all of these solutes, are coming from current photosynthesis, then the plant has to be able to photosynthesize to do that, to fill the grain, let's say. And so anything that disrupts that, like a dry soil that closes the stomata, photosynthesis goes down, solutes aren't produced, and you can't move them into the grain. So that has a big role to play too. Okay, evidence for this idea: positive pressures have been observed in phloem. Just like here, the pressure potential positive in the phloem, negative in the xylem, and that's been observed. Presence of solutes in the phloem, high concentrations, has been observed over and over again. Virus movement can be explained by this theory of pressure-driven flow. But keep in mind that the theory doesn't explain everything. In particular, why does anoxia, removing oxygen, or cool temperature, why do those affect the rate of transport? Over and above their effect on viscosity of the fluid, it seems to be the case that the flow, those cells have to be alive and they're respiring, and that rate of respiration has something to do with the rate of transport, and that's not explained by Monk's theory at all. Flow and loading, we talked about this just last time. How do the solutes get from the cytoplasm of the photosynthetic cells into the sieve tubes? It depends on the tissue, and in many plants, it involves an apoplastic pathway. So the photosynthesizing cell makes sucrose in the cytoplasm. It actively pumps that sucrose from the cytoplasm out into the apoplast. Remember, sucrose has trouble crossing the plasma lemma. It won't do it passively. The cell has to expend energy to do that. And so it does, and it pumps it into the apoplast, resulting in a high concentration of sucrose in the apoplast here, lower concentration by a companion cell, either a transfer cell or ordinary companion cell. Sucrose diffuses over to this point, and then this companion cell actively takes up that sucrose from the apoplast back into the cytoplasm. Again, it's an active process that requires energy because the sucrose won't cross the membrane by itself. Once it's in the companion cell, that results in a high concentration of sucrose in this cell, in fact, even higher than in the sieve element. And so sucrose diffuses through the plasmodesmata from this cell into this cell. Remember, plasmodesmata, open hole in the cell wall and cell membrane. No, nothing blocks that. Many plants operate this way. The energy to pump sucrose from the cytoplasm into the apoplast, or to take it up from the apoplast into the cell, is provided by ATPases embedded in the membrane. So ATP is the source of energy. Break it down to ADP, pump protons, results in an electrical potential gradient and a pH gradient across the membrane. Then sucrose proton symporters, also embedded in those membranes, allow a proton and a sucrose to come in together. So the ATPase and the sucrose transporter found in the companion cell membranes, found in the mesophyll cell membranes, are responsible for that movement of sucrose. In contrast to that, well, here's some examples of why that is. In contrast to that, cells, plants in other, I think probably smaller group of plants, particularly the Cucurbitaceae, use a different method for phloem loading called the polymer trapping model, where sucrose diffuses from the mesophyll cell through plasmodesmata, abundant plasmodesmata, to a companion cell called an intermediary cell. High concentration of sucrose here, lower concentration here, because when the sucrose gets here, it's combined with another sugar to make raffinose or stachyose or verbascose, a bigger and bigger sugar. Keeps the sucrose concentration low here, so the diffusion goes this way. And then this bigger molecule, high concentration here, lower in the phloem, diffuses through plasmodesmata into the cell. And the relative sizes of these plasmodesmatal connections may have a role to play in how big of a molecule can fit through there. That's called the polymer trapping model. That also works very well. Phloem unloading, and not so much the same. All right? That's it. That's what I think is important. Amongst the many things I think are important is what I've just said. There's a lot more to it than that.